Okay, I am going to attempt to do a little video to show you how DNA replication works. These papers, I want to give credit where credit is due. I got from a worksheet from Manitoba Fisheries. I don't know why they're teaching people about DNA, but that's a good thing. So if you want to look at their worksheet, you can go there. Okay, so this is double-stranded DNA. These are our sugar phosphate backbones here and here, and these are the pairs of bases or base pairs. So for example, A and T, G and C. I've colored them so that you can hopefully tell apart the different ones just to make it a little bit easier. Now around you'll see I have cut out other nucleotides that are just floating around. So when replication happens, the DNA will unzip and separate, like so, and the cell's machinery will be able to start finding the correct base to pair up with this one. So we've already said before that A always pairs with what? What is it? It always pairs with T in DNA. Hang on, let me find the right T. That's not the right T. Oh wait, yes, this was the right one. Sorry. Okay, so the A will pair up with the T, and what did we say pairs with G? It's C. So now that nucleotide will come along and pair up with that. There's a little machine that will bind the sugar to the phosphate. Now we need to pair up a T with its partner, which is always A. Then the A will pair with a T. This C will pair with a G. We've got one more to go. This C will pair with this G. Okay, so now we have this new strand of DNA, and at the same time this is happening on the other side in a slightly different way. And for this one I'll start at the bottom, even though I'm not really explaining why they would go in opposite directions, but they do get put together in opposite directions. So remember that C's and G's always go together. Now we need an A for this one. We need a T to go with this A. We need a G to go with that C. And we need an A to go with that T. So now if you look at the sequences of these two, they are identical to each other. And it's because of that complementary base pairing system. Okay? So because an A always pairs with the T, if you want to make this combination, it doesn't matter if you have this side or this side, right? If you have the A, you know to put the T here. If you have the T, you know to put the A here. So this is why we end up with two identical molecules of DNA at the end of this process. And they're identical to the original molecule of DNA that we had at the beginning.